Alrighty, I'm gonna get started here. Thank you all for coming. So my name is Alan Liu, and I'm an associate professor in mechanical engineering. I also have appointments in biomedical engineering, biophysics, cellular molecular biology, and applied physics. So I want to quickly introduce myself, and then we'll dive right into the presentation. So I've been at U of M since 2012. I was originally trained as a biophysicist. I did my PhD at UC Berkeley, and then did a postdoc at the Scripps Research Institute in San Diego in quantitative cell biology. And then I moved to Michigan. Currently, my group has 6 PhD students and 3 postdocs, and we work in the area of cell mechanics, mechanotransduction, and synthetic biology. So I'm going to spend a few slides in the beginning of my talk to share with you sort of what my overarching lab interest is, and then we'll specifically talk about this topic of cell-free expression in building synthetic cells. So I want to thank Marcy for inviting me to this tools and technology seminar. When I look back at the list of seminar topics in the past, I realized there was a lot of talks centered around computational medicine and bioinformatics, and I know my research doesn't quite fit in this exact category. But what I'd like to share with you is try to frame my work within this tool and technology context. So the tool I'm going to share with you is cell-free expression, and the technology is synthetic cells. So hopefully at the end of the talk, you can hopefully appreciate cell-free expression as a tool and synthetic cells as a technology. And also would, I've given a number of seminars via Zoom now, and I know it's very difficult for me to see questions at all. So maybe what we'll do is to have the questions at the end, but feel free to unmute yourself. And if there's any clarification I can provide while we're still on the slide, feel free to do that, as I cannot see any of you or see your faces or be able to read your comments in the chat box. So we'll do that. All right, I'm going to dive right in. So my lab works in several different areas. So say quantitative cell biology, synthetic biology, cell mechanics, and microfluidics. There's a common theme among these 4 areas, which is my longstanding interest in mechanobiology of biological membranes. And I'm generally interested in how forces are sensed in by living cells, and how cells are able to respond to sensing these forces. And I'll provide a few examples of why I think this is a very interesting problem. So any organisms on Earth, living organisms from bacteria to humans are mechanosensitive. So you can see examples of a C. elegans that's moving on an agar plate, that if you touch it, it reverses back. And if you go on beach, these little creatures, the animal, if you touch it, it squirts out water. Mimosa plant, which is a native plant in Brazil, also have these mechanosensitive functions. And Venus flytrap, illustrated here, also has a pretty dramatic reaction when it's simulated by mechanical forces. It's also true in our body that cell mechanics play a major role in health and disease. So if you have a injury that someone wears a cast, after the cast is removed, you have atrophy in both the bone and muscle. This is also true in condition in cardiac hypertrophy, where your heart wall is thicker, and this allows it to sort of it's for pumping blood. And in asthma attack, you have hypercontractility of the airway smooth muscle cells. So the airway becomes more restricted and harder to pass air. And finally, you're listening. As you're listening, your hair cells are being stimulated by pressure waves. And there's a linker called the tip link, and that basically stretches and activate ion channels so to stimulate the hearing sensation. So there are many processes at the physiological level that rely on mechanical forces, or rely on the roles of cell mechanics for their proper functions. As I said, I'm interested in how forces are mediated at the single cell level. So here illustrated as a cell, you can have a variety of mechanical or physical stimulus, for instance, shear stress that are applied on endothelial cells, strain forces that occur when your tissue are stretched, like for instance, as you breathe. The extracellular matrix rigidity is also an important stimulus to cells. And ultimately, these physical stimuli affect cell function. And the functions that we care about are gene expression, migration, but how does the stimuli convert a cell function? And this is mediated through these mechanosensors. So these are proteins or protein complexes that are largely on the plasma membrane, right? Receptors, mechanosensitive ion channel, cell-cell adhesions, focal adhesions, and the forces are mediated through cell skeleton network from outside the cells into the nucleus. So the highlighted are areas that my lab have actively been working on, and I'll talk a little bit about our work on mechanosensitive ion channel today. And later on at the end, we'll have a little bit of discussion on thinking about how to use cell-free expression for studying gene expression. So the work I've described so far are mostly on our cell level work, but as I said, I'm interested in synthetic biology. And for those of you that don't know, synthetic biology is a field that we're interested in building. So you can imagine, every component that you can find in a cell is a part, and we can take these parts and build complex structures. So my lab is interested in building synthetic cells using biological components, and I'll talk more about that later on. But the idea is that these parts can be used for a variety of purposes, and they can be highly modular. And if you think about building a cell, the cells have organelles, but have also compartmentalized functions. And the question is, can we build these functions out of simplified components? And synthetic biology, to most people, essentially relies on a concept of input and output. So you can imagine a cell being a computational unit, and it senses a variety of input, and molecules within the cells are processing and have respond. Hang on a second, my pointer is not working, and they can respond by providing a certain type of output. Workflow of a synthetic biologist, and oftentimes it's very similar to what you would do in experiments, right? You start with the concept, designing, modeling, simulating, probing of a cell functions of a genetic circuit. But the nice thing about biology is that it has the ability to evolve. So we can evolve, a gene of interest or something related to that we can then take this, experimental loop around. I'm having some issue with my computer. Okay, hopefully it's okay. All right. So conventional synthetic biologists often think about engineering organisms, and here just giving you examples of. My computer is a little bit frozen right now. I hope it's not. It's okay. I'm having trouble moving my pointer. Okay, there you go. So here's an example of engineer bacteria, engineer yeast cells, or even engineering mammalian cells by introducing different type of genetic circuits. So that's one type of synthetic biology, and the ones that my lab is interested in is it's a different, I will say, subset of synthetic biology we call bottom-up synthetic biology, where we actually use something called cell-free expression as the main tool. And what cell-free expression refers to is the ability to execute a genetic program outside living cells, and opens opportunities in biotechnology and biophysics. So what I quickly give you outline, so talk a little bit about transcription translation, and study using cell-free expression. And my lab does a lot of work on membrane proteins, and I'll show you some examples of that. And then we'll talk about synthetic cells, which is using the cell-free expression system as the material that are within our cell, just like how your natural cells, there's a membrane around it. And then, I have a single slide on thinking about cell differentiation that may appeal to this audience. So diving in cell-free expression has been around for a long time, and essentially it's a couple reaction of transcription translation, or we call TXTL. So if you add a piece of DNA either in plasmid form or linear form, along with a mixture of cell lysate, and you have a metabolite. You can put this in a compartment. You can essentially run this transcription translation reaction. And this can be done in a variety of volume formats. So at a large scale, you can use it for industrial purposes of making biologics. and you can run them for prototyping at the levels of well plates and test tubes and even paper. And we work in the areas of encapsulating them in these little tiny compartment made of lipids. So basically, these are lipid bilayer vesicles or liposomes, and we use them as models of artificial cells. And just to give you a few examples of how they've been used, and I'll be very quick on these applications. And these sulfur expressions are essentially lysates from cells, and they can be dry on a piece of paper. And if you then hydrate them, you can incorporate genetic circuits on these paper. Once you freeze dry them, you can rehydrate them. So they've been used for as reporters. So reporters of viral DNA, and the work out of Jim Collins' lab at MIT showed that a number of years ago. So as an example how this sort of works, so you can put on a genetic circuit. This is an inducible circuit that is activated by tetracycline, and that drives either GAP or mCherry expression. And you can see that this is a paper disc, roughly about 2 millimeters in size. And if you add tetracycline, you can see GAP gets made, or mCherry gets made. And if you don't have tetracycline, the paper discs remain non-fluorescent. So, so the workhorse of these cell-free systems essentially are just lysates, right? So in biochemistry, this has been used a lot, and sort of it essentially bypasses the challenges of purification, right? So normally, if you want to work with a recombinant protein, you would grow up a liter of a bacteria after you transform them with the plasmid interest. You will isolate them by breaking the cells apart and purifying them through chromatography. It might take a week or two to get the proteins. In cell-free protein synthesis, essentially you still carry out the cell lysate, but these cell lysates are treated in such a way that you can essentially have these lysate be stored, okay? So you just put them at -80. And then when you're ready to do the experiment, you simply supply them with the energy solutions along with DNA, and then you can make the protein. And it will take much less time, obviously, from the start to finish, because you don't really need all these other steps. So that's essentially what we do in the lab, is to take mammalian cells we lyse, and I'll talk about that. But like I said, this technology has been around. This tool has been around for a long time, and it was essentially what was used to study to decode the genetic code 50, 60 years ago. But it's also been used very heavily for biotechnology in terms of making proteins or enzyme of interest. And this is just a quick timeline to show you the different type of proteins that have been made and the concentration that came along with it. But my lab is interested in using cell-free expression systems to make membrane protein. But I would say generally that technology or tool like this can be utilized in a variety of contexts to study, different enzymes or proteins. So what we use in the lab is a mammalian system mostly. So we work with bacterial cell-free system, but also mammalian cell-free system. And the mammalian system we use is based on helolysate. And we also supplement with 2 other enzymes. These are soluble fragment of an enzyme called GAD34. And that basically promotes translation by dephosphorylating the initiation factor 2 alpha. We use exogenous transition factors. So here we use T7RN polymerase. And so any DNA that we have has to have a T7 promoter. And then we just simply mix other buffers, right? So they will contain NTPs and amino acids. So if you do this type of reactions on a plate reader, you can see this is making GAP, and we can get a very nice production of GAP over time. If you don't have plasmid DNA, you get absolutely nothing on for an experiment like this. So just I'll have a couple quick summary of studies we've done in the past to show you what we can use this for. So this is an example of looking at transcription and translation simultaneously in a droplet or vesicle format. So here, we make use of this, locked nucleic acid. So this is a synthesized, probe that has a quencher and a fluorescent dye at the end that's already chemically attached. And in the presence of a target piece of RNA, this will actually displace, because this piece is longer, it will displace the one of the strands and basically separate the quencher from the 4,4. And now the 4,4 can fluoresce. So if we run the cell-free expression system, the idea is that the RNA or the mRNA as it's being produced, it can be detected, by our LNA probe. And the protein is fluorescing on its own. It's a GAP, so we can see the fluorescence in real time. So if we do these bulk measurements, so here's with the different amount of DNA input, we can measure the amount of mRNA that's being produced, right? This is the M-cherry. This is the 4,4 attaching to the mRNA. So we're measuring simultaneously mRNA production and then using GAP fluorescence as a metric for protein production. So you can see that there's a time lag of translation, as we would expect, but the transcription happens pretty quickly. So this is a measurement that we do in bulk. But we can also do this in a compartment of a droplet. So we use microfluidics to generate these single motion droplets. So these are water in oil droplets, and we encapsulate within that the cell-free expression system. And if we don't include a probe, the LNA probe, we do not measure any mRNA. We can obviously see the proteins are synthesized. If we include the mRNA probe, we see the mRNA fluorescence in the M-cherry channel, so we know mRNA being made, but we also are making proteins. So the transcript dynamics is very different between bulk and so and then the experiment we did is to look at, as a function of time, what is the amount of mRNA made. Actually, what's interesting is that the mRNA decreases over time. Okay, so if you remember the bulk study, that continued to increase over the time course, but in the confined system, the mRNA is actually decreasing while the protein in green is increasing over this period of 5 hours. So we didn't carry out the study in great detail, but I think just from these two examples, you can see that the transcript dynamics is actually very different between the bulk assay versus a confined system. So I'm going to switch gear and talk. The second part of this CAB part is to show you some work that we've done on membrane proteins. So we have a lot of interest in working with membrane proteins as I think they're really important in mediating any communications between outside of the cell to inside of the cell. So for this project, we looked at a membrane protein that is that's also very important in my interest on mechanosensitive. And this protein is called SUN protein. They're protein that resides in the nuclear membrane envelope, and essentially it mediates the mechanical communication between outside the cells and inside the cell. So the inner part of the SUN protein binds to lamin. Okay, so in the C-terminus side has the SUN domain that binds to cache peptide. And these cache peptide proteins are called nesprins, and they basically connect to the cell skeleton. So it forms a mechanical linkage from outside the cell to inside the cell. So this is what the link complex stands for. And the SUN proteins have the SUN domain and cache domains. So this protein's been known for a long time, but it's really difficult to study them biochemically. And people who study these proteins, who do them in cells, are constantly challenged by the fact that it's a nuclear envelope protein. The nuclear envelope localization makes it hard to study. But if you try to do this biochemically, no one has purified the full length protein yet. So instead they work with soluble fragments of either the luminal domain, which is a SUN domain, or things that happen on the other side. So we wanted to see if we can use cell-free expression system to actually make full-length SUN proteins. And traditionally, making membrane proteins is very hard. So from a biochemistry standpoint, these are generally purified as membrane protein in these small vesicle proteoliposomes, and then they can be dried down onto a film, and then rehydrate them. And this is a process called electroformation. Once if you do these hydration under electric field, you can make these giant vesicle that have the membrane proteins. You can also do the same thing on hydrogels, and you essentially achieve the same reconstitution of membrane protein into the lipid membrane. So I won't go into details of the proteins because we'll talk about that later, but what we want to use is the cell-free expression, right? Using in vitro transcription translation to essentially get these membrane protein into the bilayer. And this particular protein is called MSCL. Again, that will have a few slides on that later on. But we wanted to essentially bypass the purifications of these proteins, and MSCL is able to integrate into the membrane. And again, this slide will show up in a little bit. So with that idea in mind that we wanted to use cell-free expression to reconstitute membrane proteins, we devised a very simple assay, and this is based on support lipid bilayer. This is called super template, so support lipid bilayer with excess reservoir. And you have these 5 micron glass beads that can be deposited with a lipid bilayer on it. And we add our cell-free expression reaction, incubate them for some amount of time. And the hope is that we get these membrane protein incorporated into the bilayer. So now you can, if we go back to the schematic of these some proteins, we have the N-terminus, in which we will tag with the EGAP, and we have a C-terminal sun domain. And we know there's some transmembrane domain that is important for them to be in the bilayer. And there are 2 possibilities for insertion. So we're interested in seeing if we can use the cell-free expression to understand topology of these membrane proteins. So when you reconstitute a membrane protein, one possibility is that this internal domain will be inserted into the side, basically, that's protected from the external solution. So that if we add a protease, then we'll digest the sun domain, but we will not be able to digest the EGAP. So EGAP is protected, as this cartoon illustrates. The other possibility is if the insertion is opposite, right? So that the N-terminal is facing outward. In that case, then protease digestion will basically cleave the GAP. Then you have no fluorescence. Okay, so there are 2 possibilities. So we wanted to test that. And first of all, before we do that, we want to make sure we can actually get some protein in the bilayer. And indeed, if we have super template, we express our SUN1. So I forgot to mention there are a couple isoforms, SUN1 and SUN2. So both full-length SUN1 and SUN2 are able to incorporate into the support lipid bilayer on the super template. And if you don't have the bilayer, it doesn't go on at all. And what's interesting about SUN1 and SUN2 is that previous predictions have shown there are actually a couple transmembrane domain for SUN1. And there's 1 transmembrane domain for SUN2. Now, if you have a membrane protein that needs to have its N and C termini on opposite side of bilayer, it's impossible to imagine having 2 transmembrane domain because it will fold itself back so that N and C termini will be on the same side. So this was a little bit confusing, even in the literature, as to how this topology can actually work. But what we did is to first of all test the insertion itself, and if you see using a purified GAP with this protease assay, we can eliminate fluorescence within a few minutes. Basically, all the fluorescence completely gone because we add the GAP on the outside of the membrane in the bead, and the GAP is completely digested. Now, when we look at full-length SUN1 and SUN2, you can see very easily that the GAP is not basically cleaved at all by the protease, so that they're protected from the protease. That means the GAP, the internal GAP, is facing inside the bilayer, whereas the SUN domain is exposed to the solvent. Okay, so this is quantified over here just for the SUN1. To give you an example. So what so this result shows that the C-terminal SUN domains are actually solvent exposed and exposed to the which is the luminal side of the protein. So that's good. So we can reconstitute these some proteins. There are the topology appears to be 100%. All right, and there's reason because this is made biosynthetically rather than being purified biochemically. And then, so we want to get back to these questions of why we can have them on opposite sides. So the top is the original, essentially the original predictions, but then we went into a different prediction software and found that there are a couple more, basically a couple more hydrophobic region. But before we did that, we wanted to see if the N-terminal domain and luminal domain can associate with membrane. Okay, so we essentially chop off where this transmembrane domain is, and generally a nuclear domain only construct and a luminal domain only construct. And you can see that in the luminal domain, in the nucleoplasmic domain, construct, we still see association with, with the membrane. And so this suggests there's something within this construct that actually has membrane association capacity, right? Whereas the luminal domain is completely soluble or complete cytoplasmic and doesn't really bind to the membrane. Okay, so as I said, it turns out that there are two additional homology hydrophobic regions that exist within this nucleoplasmic domain, and we generate a couple more mutants, taking one of them out and taking another one of them out, and then we wanted to see if these are still binding, right? So when we first generated, so this 1 to 364, this one has one hydrophobic region, and this again binds to the lipid-coated beads. Now, if we cut that one off and generate this 1 to 232 mutant, and this one is completely soluble. So we know that there are, based on this results, we can see there are a couple hydrophobic regions that are potentially still present on SUN1 protein. And so to know the topology of that, we basically turn to our protease assay, protease digestion assay. So if we look at the nucleoplasmic domain, it associated with the bilayer, and if we added the pronase, it doesn't digest. And if we do this, the same thing with cutting out one of the hydrophobic region, again we do not remove the GAP. So based on that, and if of course if you truncate again, then that goes away. So if you piece all this information together, you will we essentially came out with the topology of a SUN1 protein where the original transmembrane domain in yellow basically marks over here, right, where you have 1 and 2, and then we identify two more hydrophobic region, and one of them is a peripheral associated hydrophobic region, because if we added, because we know that's the only way it can work, and the third one is to basically bring it through the bilayer, and this will allow us to have the EGAP on the nucleoplasm side, and then the SUN domain are luminally exposed. So so this is sort of a tool, I guess, thinking about self-expression as a tool allows us to study I would say a difficult transmembrane protein. So last, quickly to highlight the fact that this is a functional SUN protein, is that we can take a cache peptide. So remember I said the SUN protein can bind to cache protein, and there's a stretch amino acid that is known as a cache peptide that can bind to the SUN protein. So we synthesized this cache peptide and added a fluorescent label. And there's 4 amino acids that are critical for binding to the SUN protein, or to the SUN proteins, and we made a mutant cache peptide. So you can see the wild type cache peptide binds very nicely to SUN1 and SUN2. Now, if we use the mutant peptide and really just lacking the 4 critical residues, we completely eliminated binding of these cache peptide to the reconstituted SUN protein. So so we're very excited that we can make a functional full-length SUN proteins and are still able to bind to cache peptide. So that completes sort of the talk on the cell-free expression as a tool. So I'm going to switch gear and talk more about using cell-free expression in the context of synthetic cells as a technology. So so this is a slide on my PhD work where I actually built a very simplified system to study actin dynamics. So I basically assemble a dendritic actin network on a giant ulnar vesicles and study how actin cell skeleton can self-assemble and deform the lipid bilayer. So at the time when I finished my PhD, I was interested in reconstituting all sorts of different processes. And these are all the processes in the cell that has been reconstituted by purified proteins. I don't work on any of these, but just to collect them in a single cartoon. And had the idea that perhaps we can use in vitro reconstitution to create all sort of cell functions that we know can be reduced into component parts. But at the same time, or around the same time I was going to start my lab, I encountered a article by Petra Schwele at the Max Planck Institute, who published a really interesting perspective article that basically talk about how we can take different components from a cell, Okay, so enzymes, cell skeleton, motor proteins, DNA, and we can construct them into a synthetic cells, or into a simplified system to study cells. So so from that, came up with the idea that can we assemble biological components in a meaningful way to build artificial cells that would mimic certain aspect of the cell. So this is something that my lab has been interested in for a number of years now. And just to put all that sort of ideas into a single slide, and this is something I'm really passionate about and really interested about, is to build a cell-like system. So this would be a compartment. So right now we mostly use a phospholipid bilayer that has molecules inside and molecules that are membrane proteins that are outside. And I say membrane protein reconstitution is one of the most challenging part of this type of work, but we have again the technology, the tools to do this now. And I want these. I would like to make these cell-like system to sense a variety of inputs like forces, molecules of light, and then produce outputs that are similar to what the cells would do, right? So be able to change shapes, right? So cell migrating or enzymatic activity or secretion. For the last many years, my lab has been working on trying to get these synthetic cells to sense a force and then be able to secrete something. So the question is, can we build an artificial cell that couples a mechanical input to a biochemical output? So this is a concept. So we have been working on trying to make artificial platelets, and I won't go into details of why we are interested in making platelets, but the idea is that making a synthetic cell is a modular approach, right? So I mentioned at the beginning that these synthetic biology uses parts that can be very modular. So in ideas of creating this artificial platelet, the four modules that need to come to this type of artificial platelet requires us to be able to figure out how to make vesicle attach, how to encapsulate, all right, cell-free expression system, and I already talked about making cell-free expression system. And then importantly for this purpose is we also require vesicle fusion in order to reconstitute the functions of what I think could be a artificial platelet. So I won't go into details of this idea, but just to give you a sense of how this bottom-up approach works is by building these modules. So the tool that we use for here, it requires microfluidics and to encapsulate. So this is a device that's made out of glass that has a middle phase. This is forming something called double motion, so water in oil and water. The inner phase is an aqueous solution, and the middle phase is an oil phase, and the outer phase is another aqueous phase. So we make these droplets. And here's a still image of what that looks like, and you can see these water double motion comes out from the from this side and basically get collected on the right hand side. And we can use this technology to make lipid bilayer vesicles that in fluorescence you can see side views and top view. And then we can, and this is examples of, if you use different composition of lipids, you can make phase-separated vesicles that have, fluorescent lipid on one side and the other side is dark. So I want to share a couple, quick data on, what we can do in terms of making synthetic cells that are mechanosensitive. So earlier I sort of intentionally skip over part of the data there, but I can talk more about this now, is that this protein called MSCL is a really fascinating protein. It's a protein that is made in E. coli. It's a homopentamer that opens a pore with tension. So if you apply a force onto membrane, opens a physical pore about 2.5 nanometer in size. And that pore allows small molecule to pass through this membrane protein. And bacteria use this because when there is a big change in osmotic pressure, so after rain, the osmolarity outside basically goes down, and you have water coming into the cells and increase the sugar pressure. If the pressure increases too high, then the bacteria will burst. So it turns out that these MSCL, these mechanosensitive ion channels are important for opening so that it can equilibrate osmolyte. So it can equilibrate osmotic pressure across the bilayer, okay? And this is a survival mechanism for bacteria. So my lab has some work in the past few years of taking this bacterial protein and put them in mammalian cells, and I think it's really fun to work with these channels in mammalian cell, because it's almost like a mechanogenetics type of experiment, right? So instead of using ion channel that's sensitive to light, we're using ion channel that's sensitive to force. But anyways, that aside, we were able to reconstitute MSCL into bilayer vesicles. So general idea here is to make a vesicle that has, with our self-expression system and having this membrane active property. And then we also at the same time wanted to have a biosensing activity. So for biosensing, we're using a fluorescent biosensor called GGECO that is sensitive to calcium. So we have a couple genetic circuit, one that makes MSCL, and then that one also makes this calcium biosensor, okay? I guess the exact construction of these two circuits is not that important, but the idea is that we can make two proteins at the same time, one for detecting calcium and the other one for sensing the osmolarity so that it can open a channel. So when we take this, we had a different approach to make a vesicle. So if we're able to reconstitute this MSCL in a bilayer vesicle using the bacterial self-expression system, we can actually ask, can this channel be activated by transferring this vesicle to a hypoosmotic condition, right? So remember, hypoosmotic condition increases membrane tension. So if we encapsulate a fluorescent dye, the increase in tension can open the channel, allowing the dye to escape. So this is the data I skipped over earlier. So if you don't have MSCL, right, the dye that are encapsulated in the vesicle basically is retained over time. However, if you express MSCL, and this is in the hypoosmotic condition, that the fluorescent dye would escape. Okay, so this is a very simple assay to look at the functions of MSCL. So we are able to produce a synthetic cell that is now mechanosensitive, that when that's under hypoosmotic condition would open the pore. So now we're ready to look at the GGECO, right? So the GGECO is a calcium biosensor, right? So when in the presence of calcium, the GGECO will be fluorescent. So if we encapsulate GGECO along with calcium, so we'll see that the vesicle is green, as you would expect, because they're co-encapsulated. Now, if we put the calcium on the outside of the vesicle and have the vesicle express GGECO, then the vesicle will be dark because there's no calcium for GGECO to fluoresce. And then if we express MSCL and GGECO at the same time, if the solution is isoosmotic, then we have no fluorescence. But if we put the vesicle in a hypoosmotic condition, then you will see that the vesicle is fluorescent. Okay, so because the calciums are able to enter the vesicle through activated MSCL, mechanosensitive channel. Okay, so this is a quick demonstration of a artificial cell with an AND gate property. So AND gate, in computer science and logic, is basically where you have two inputs are both present, right? So here the synthetic cell is sensing a mechanical input, which is osmotic pressure, and a chemical input, which is external calcium. If both of these are present, then the vesicle will be fluorescent, right? So we went on further to basically add a inducible circuit. So we have a circuit that is very commonly used in recombinant protein synthesis is the lac operon, and that needs the inducer IPTG. IPTG normally is impermeant to the cell, right, or to the vesicle, but we also express MSCL. So if the MSCL is activated, then IPTG can enter. And as the downstream reporter protein, we're using a homolog of actin called MREB, and this is a actin homolog from bacteria. So if you co-encapsulate IPTG along with the self-expression system, you can see a nice ring, a cortex formed by MREB. If you have the IPTG in outside of the vesicle, there's no fluorescence at all. Now, if you express, if there's no MSCL, okay, and have a hypoosmotic condition, there's no ability for IPTG to enter the vesicle, so there's no fluorescence at all. But if you have a hypoosmotic condition with MSCL, then you see a MREB ring again. So MREB has a tendency to self-assemble at the membrane and is used in cell division for in bacteria. So we found in this work that osmotic shock can lead to the expression of MREB that assemble into a cortex-like structure. So with that, so I'm going to give a quick summary and then basically have this one discussion slide that I think may be of interest to some of you. So talk about today the use of self-expression as a tool, right? So the self-expression can be used to study transcription and translation dynamics, and they're used very heavily in the field to prototype genetic circuits. I've also talked about using it as a tool to study membrane proteins. We can use it to reconstitute membrane protein and be able to study the topology of it. As a technology, we use self-expression to make these synthetic cells. This bottom-up synthetic biology provides a ground for making these synthetic cells. We have made these mechanosensitive synthetic cells that are able to respond to osmotic pressure and open a pore and allow external molecules to enter the synthetic cells and activate gene circuit. So those are some of the topics I spoke about. So I want to end with sort of idea and just to get people's thought on this is, given the potentially high throughput nature of self-expression, right? We can make we can take lysate from cells and we can freeze them down. We can allocate them to small volumes and so on and so forth. So they can potentially be used for high throughput strategies. Can they be more broadly used in basic biomedical science as a research tool? So I know this crowd, a lot of folks interested in bioinformatics and perhaps in sequencing technologies. So wonder, as a discussion point, it can be used to study stem cell differentiation. So each of us, a human being, have lots of cells in our body, right? So a cell, single cell becomes 200 cell types in our body, roughly 35 trillion cells, and we know. Stem cell differentiation, really there's one set of there's one genome, but depending on what is being expressed, it can actually be going to different cell types. So can we consider the possibility of taking, let's say, isolated nuclei, and this is in the context of aging, but you can think about it for studying just stem cell differentiation itself or even cancer. But the idea is, can we take isolated nuclei? And we know that there are many transition factors that are involved in turning genes on and off. Can we use self-expression system to produce combination transition factors? Okay, so here I'm just listing 1 to n, and I believe there's a library of transition factors that are known. We can probably clone them in our self-expression vector and basically just make these transition factors by self-resynthesis, and then incubate them with nuclei, and then perform some type of sequencing analysis on the transcriptome, or let's say ataxia, and what, just sort of using these pretty I would say emerging or analysis to understand what which part of the genomes are being activated and what transition factors are responsible for that. So I think perhaps open up a possible venue for doing something sort of different, right? Using self-resynthesis. So that's essentially my one slide. I wanted to get people's thoughts on that. But let me close by thanking my group. So the work I presented mostly come from Sardi Majunda, who was a former PhD student and staying on my lab as a postdoc for a year, and some of the work on this on the simultaneous monitoring of transcription-translation comes from a former postdoc, Xu Wang, and the work I presented had been a collaboration with Ann Luxton and Vincent Nero, both at Minnesota. Right. So with that, I'll stop and take any questions.
You had one in the chat. If you
yeah. Let me escape out of this. So I need to first go back to arrows. Okay. So let me see if I can view that. All right. Perhaps this is too far outside the scope. All right. So I'm just going to read the question now as well. So since I have not seen the questions, Do you have thoughts regarding using the cell-free system to test properties of proteins to be involved in phase separation? Interesting. The challenge of identifying the what and how of selectivity for subcellular compartment seems like could be addressed using tools you discussed. Oh, interesting. So I have not thought about the exact idea. So the phase separation right now is obviously a very hot topic in cell biology, and these are, a lot of it is coming from these intrinsically disordered protein or through multivalency. So I happened to do my sabbatical a couple of years back and worked on phase separation at the time. But I'm not, I have actually used cell-free expression to make that protein and that phase separated proteins that can be tested in vitro. But we have, currently in my lab, we have not used it for this purpose, but I'd be pretty curious to see, how we can take, cell-free expression into different domains. So certainly a interesting idea. Yeah. So I think the nice part about cell-free expression is you can make any protein of interest that is not, that may not exist in in the original, I would say, host cytoplasm, right? Because and there's also no additional transcription going on if you don't provide, since we're providing a T7 RNA polymerase, and there's no nuclei, there's no other, transcription. There are no other polymerases in there. So. So we have not done it exactly on phase separation, but it's interesting idea. All right. Okay, so maybe I'll stop sharing. Maybe that's easier to. All right. Well, I'd be very curious if anybody here had thoughts on the last slide on this, perhaps using this for studying differentiation. Not so viable, but the idea is to look at different combinations of transcription factors. Yeah. Yeah. So from Dana again, that likely challenges how well characterized the transcription factor are for a system. That absolutely true. Yeah, I was able to find I think University of Toronto had a researcher from there had a paper in Cell a couple years back, basically cataloging all the transcription factors, but I'm not sure if all of them are well characterized or well studied. But I also worry about post-translational modifications of these transcription factors, whether they're necessary for their functions, right? So that's something that probably will not be easy to reconstitute. But I was inspired by just sort of how iPSC cells are generated, right? So they basically are just, transducing 4 transcription factors, and they can turn cells into iPSC. So sort of that's where a naive idea coming out from is just to express transcription factors in the cell-free system. Yeah. Right. So so it's probably true from Dana again that most of the transcription factors are probably not well characterized, and perhaps it could be a platform to first of all study these transcription factors and can characterize them in the vitro context. Yeah. So. Great points. Thanks, Dana. Yeah. All right. Are there any other questions or thoughts, comments? All right. If not, well, we're right on time. Well, thank you all, and it was a pleasure to share this work with you. And if you have any questions, feel free to reach out to me, and be very interested to chat
Thank you for your talk, Alan. That was great.
Oh, yeah. Thanks, Aaron. Yeah. All right. And yeah, have a great rest of the semester, guys. All right. Thanks. Thank you, guys. Yeah.